This lecture covers Chapter 26, Being Multicellular. As we've discussed previously, physiology is the study of the processes and functions of an organism. The field focuses mostly on complex multicellular organisms, so we will first focus on what it means to be multicellular, the evolution of complex multicellularity, and then some of the advantages and challenges to living this way. Of the 119 major groups of eukaryotes, 83 contain only single-celled organisms. Cells eat other microorganisms or ingest small organic particles, live suspended in water columns, or are parasites living within other organisms. The 36 remaining groups exhibit at least some cases of simple multicellularity in the forms of filaments, hollow balls, or sheets of little differentiated cells. There are some common properties of multicellular organisms. These include cell adhesion molecules that cause adjacent cells to stick together, little communication or transfer of resources between the cells, little differentiation of specialized cell types, and as a result, most of the cells retain a full range of functions, including the ability to reproduce on their own. Lastly, nearly every cell is in contact with the external environment. 6 groups of eukaryotes have developed cenocytic organization. This organization involves the cell nucleus dividing multiple times while the cell itself does not divide. The resulting multinucleated cell can be large in size, as shown in these 2 common marine algal species. The vast majority of biota that we see around us are complex multicellular organisms. These include land plants, animals, red algae, brown algae, and fungi. Plants and animals are the complex multicellular taxa that we will focus on in this course, so we'll talk about them in a bit more depth. Plants and animals differ in many ways, but they have some fundamental features in common. For one, they have highly developed mechanisms for adhesion between cells. They also have specialized structures for cell communication. There's quite a bit of tissue and organ differentiation in their bodies, and typically a very small subset of cells contribute to reproduction of the organism. In these multicellular organisms, cell or tissue loss can be lethal, and they also have both interior and exterior cells, so that only some cells are in direct contact with the environment in which that organism lives. Let's take a look at the overall phylogeny of eukaryotic organisms to see the places where complex multicellularity evolved. You can see that it evolved independently in several different lineages. To orient you to this figure, the clades labeled down the right side are referred to as supergroups in the eukaryotes. You can see that there is complex multicellularity in 3 of these groups: the opisthecons, archaeoplastids, and stirmenophiles. Among the opisthecons, multicellularity evolved in animals and twice independently in the fungal group. Among archaeoplastids, multicellularity separately evolved in the red algae and again in the green algae, which includes land plants. Finally, multicellularity again evolved in the brown algae. So we have 6 different times in Earth's history in which multicellularity evolved. All organisms must obtain nutrients from the environment and eliminate wastes back into the environment. Diffusion and bulk transport are 2 ways in which organisms do this. Diffusion is simply the movement of molecules from an area of high concentration to areas of low concentration. A common analogy for this is that of a spray air freshener. The spray emerges from the bottle in a small area and then spreads outward to areas of lower concentration. Bulk flow, on the other hand, is the means by which fluid pressure is used to move molecules through organisms at rates and distances beyond those possible by simple diffusion alone. Bulk flow is seen in animals when pumping blood through the circulatory system to deliver oxygen to and remove carbon dioxide from tissues that are at great distances from the lungs. Other complex organisms also rely on bulk flow. For example, trees must transport water upward from their roots to their leaves and sugar downward from leaves to other parts of the plant. An important consideration when comparing single and multicellular ways of life is the surface area to volume ratio. In this example, we have two cubes, which differ in surface area by a factor of 25. If we compare their volumes, they differ by a factor of 125. The larger cube, therefore, has a much smaller surface area to volume ratio. Similarly, larger organisms, such as multicellular organisms, have a much lower surface area to volume ratio than do smaller organisms. This means that less surface area per unit volume is available for exchange with their environment in the larger organism. So, for a larger organism, diffusion is much less effective as a means for obtaining nutrients from and eliminating wastes into the environment. Diffusion is effective only over small distances, so it strictly limits the size and shape of single-celled organisms such as bacteria. Diffusion also constrains how eukaryotic organisms function. Therefore, some large animals have developed various mechanisms for circumventing the limits imposed by the diffusion. On the left here, we have a sponge. The large size of a sponge is made possible because the large metabolically active cells are in close contact with the environment. This is enabled by the use of canals in their bodies that move seawater past the metabolically active cells. The jellyfish on the right also has thin layers of tissue that are metabolically active, but the bell shape on top is thick because it is mostly full of inactive molecules that do not need to be in close contact with the environment. There are three general requirements for complex multicellular life. First, cells must be able to stick together to build a multicellular body. Secondly, these cells must communicate with one another. And these cells must also have a genetic program that guides their coordinated growth and cellular differentiation. Over the next few slides, we're going to look at the first two characteristics in a bit more detail. Complex multicellularity requires adhesion between cells. In animals, adherins, integrins, and some additional transmembrane proteins, that is, proteins that cross the cell membrane, provide the molecular mechanisms for adhesion. In plants, the adhesion molecules are carbohydrates called pectins. Pectins are the molecules that make fruit jelly gel. So what evidence do we have about the possible evolution of cellular adhesion? For this, we can look to the closest protistan relatives of animals, unicellular microorganisms called choanoflagellates. Their genome has genes that code for many of the same protein families that promote adhesion in animals. Although they are generally unicellular, simple multicellular aggregations can be induced by molecular signals in a number of these choanoflagellate species. The left-hand image in this figure shows the typical free-floating nature of these organisms. The central image shows an individual cell forming a stalk to adhere to a surface when the cell contacts it. In the right-hand image, we can see how multiple cells can form multicellular aggregations when contacting each other. So how do these choanoflagellate cells adhere? The genome of these organisms code for some of the same adhesion proteins that are found in animal cells. Interestingly, they form these aggregations in the presence of their bacterial prey. Scientists therefore hypothesize that these aggregations increase individual success in capturing prey. This suggests that the first multicellular animals made use of pre-existing adhesion proteins that originally functioned to facilitate feeding aggregations. Cellular communication is also important for multicellular organisms. Communication is achieved by molecular signals. Generally, a signaling molecule synthesized by one cell binds with the receptor molecule on the surface of a second cell. This can either turn on or turn off genes in the receptor cell's nucleus. These signaling pathways have been found in animals, plants, and some protistan relatives. In animals, as shown here, gap junctions are protein channels that allow ions and signaling molecules to move from one cell into another. These can help cells communicate with their neighboring cells. In plants, plasmodesmata allow the same type of cell communication as do gap junctions in animals. Plasmodesmata are intracellular strands of cytoplasm that extend to neighboring cells. Other complex multicellular organisms also have channels that connect adjacent cells, Since similar channels do not occur in most other eukaryotic organisms, they appear to represent an important step in the evolution of complex multicellularity. Complex multicellularity involves the genetic programming of all cells so that they differentiate in space. Cells have different fates depending on which genes are switched off or on by the molecular signals they receive. Individual cells are able to receive these molecular signals because they have plasmodesmata or gap junctions. Plants and animals are the best known examples of complex multicellular organisms, yet the study of Plants and animals are the best known examples of complex multicellular organisms. Yet the study of phylogenetic relationships makes it clear that complex multicellularity evolved independently in the two groups. Both plants and animals have evolved sophisticated systems that cause adjacent cells to adhere to each other and that promote the targeted movement of signaling molecules between cells. The plant and animal mechanisms must differ in some way because plant cells have cell walls and animal cells do not. The plant cell wall imparts structural support to cells and provides the mechanical support that allows trees to grow tall. A trade-off is that it is also responsible for the inability of plants to move, which has two major consequences for development. For one, the entire program of growth and development involves cell division, cell expansion, and cell differentiation. The mechanical consequence is that plants' growth is confined to meristems. Meristems are the actively growing cells at the tips of stems and roots. Secondly, plants have had to evolve a variety of mechanisms with two main goals in mind, one being to transport water and nutrients without the use of energy from the soil to leaves. And since plants cannot flee predators, they've evolved mechanical structures such as hairs and spines and toxins to keep from being eaten. In contrast, animal cells are not constrained by cell walls like the plant cells are. Therefore, animal cells can move relative to one another. In the typical animal development process, fertilized eggs undergo several rounds of mitosis or cellular division, forming a ball of undifferentiated cells called a blastula. Blastula cells migrate, becoming reorganized into a hollow ball that folds inward at one location that forms a layered structure called the gastrula. Gastrula formation brings new populations of cells into direct contact with one another, inducing patterns of molecular signaling and gene regulation that begins the process of growth and tissue specification. Gradients in signaling molecules define top, bottom, front, back, left, and right. The emphasis in this presentation has been on cell adhesion, modes of communication between cells, and genetic programs that guide growth and development for complex multicellularity to develop. Not only are these three required for development of complex organisms, they had to be required in a specific order. When all these features are placed onto phylogenies, they show both the predicted order of acquisition and the tremendous evolutionary consequences of complex multicellularity. In these two figures, when we compare the evolutionary paths of animals and land plants, we can see that the first characteristic to develop with the furthest back common ancestor is adhesion and cell signaling in both lineages. We also see next the development of communication mechanisms. In animals, that includes the gap junctions, and in land plants, that's plasmodesmata. The next characteristic to develop is bulk transport. In animals, this occurred at one point. In the case with plants, we can see that bulk transport evolved two separate times.